formation there is very little difference, and this difference is with respect to its cell wall. Okay, and this difference is with respect to its cell wall, bacteria and molecules. Otherwise, many things are similar. Most of the things are similar. So today we'll be discussing the general morphological characters. CR, Datta,
Yes, sir.
and Jyoti. Okay.
Yes, sir.
So what you do, tomorrow is our college foundation day. Since you are not here, otherwise you could have also might have participated in it, 18th August, because this college was established, you know, on which date? Is Holy?
Sir, 18th August 1908.
1960.
Oh yeah, who told full marks?
Sushant
Who told? Sasan?
Kumar.
Sushant Kumar, yeah, right. It is 1960 and date was 18th August. Okay. So tomorrow is the foundation day. This 1908, someone told it is the Sabor. Okay. College, that is College of Agriculture, Sabor. It was established during the British regime. So tomorrow is the foundation day. I may have some work, so that is why, Sia, you just knock me at 4:30. Okay?
Okay, sir.
Okay. So just we'll start now. Just a minute. So you might know these microorganisms are two types. One is prokaryotic, another one is eukaryotic. In between, if somebody has some doubt, you can ask. So these bacteria are, of course, these are the prokaryotic microorganisms. Okay. So these are the prokaryotic microorganisms having 70S as ribosomal unit. Okay. S is the sedimentation coefficient. Okay. Then nuclear membrane, it is a of bacteria, and also the organelles are not bounded by the membrane. That means these organelles inside a bacterial cell, these are naked. So if we go to the definition, bacteria are extremely minute. Okay. Then rigid, essentially unicellular organism. Okay. So slide is very clear, I think. CR?
Yes, sir.
It is visible.
Slide is very clear. It is black and white. You know, is it very visible?
Yes, sir. It is visible.
Okay. You can see the bacteria are extremely minute. Extremely minute means you can't see it through its naked eyes. Okay. You can't see. Then it is rigid, essentially unicellular organisms. These are free of true chlorophyll and devoid of any photosynthetic pigment. That means these bacteria cannot produce their food themselves. Okay. So it is a very common character of organism. Most commonly multiplying asexually, both sexual and asexual reproduction is there. It is present, but generally most commonly they multiply asexually by simple transverse fission. So through fission, can you say in how many minutes a bacterial cell get divided? Average time?
20 minutes.
Yeah.
20 minutes. 20.
Yeah. Who told? Second voice I could not recognize. Sushant Kumar. Earlier you told the college establishment day.
Yes, sir.
Yeah. So you are from Sushant Kumar?
Sir, Supol, Bihar.
Oh, Supol. So there is, I think, lot of water present in Supol. It's not flooded.
Yes, sir. Yes, sir.
Very difficult, huh?
Not my area, sir, but some area filled with water.
And it is raining also, na? More or less every day.
Yes, sir. So much rain.
Anyway, enjoy rain. What to do? Not do. So generally, these bacteria multiply asexually by simple transverse fission. All these things you might have read in your high secondary. The resulting cell being of equal or nearly equal size. Okay. sorry, size of in general the bacterial cell size is 0.2 to 1.5 micrometer in diameter. Somebody is joining, is the problem. I think somebody is joining again. You see. That is the problem. If I concentrate on class, in between somebody is joining. Even someone is joining. Here,
Yes, sir.
from next class, you just send the link so that you will be admin and you will go on joining. Okay, otherwise it will disturb the class. You understand?
Okay.
Good. Anyway, so this size is 0.2 to 1.5 micrometer in diameter, and since these are the generally these are the rod-shaped bacteria, okay, of course there are focus also. The length is 3 to 5 micrometer. Okay, so already I told you these are prokaryotic. Okay, prokaryotic is primitive than eukaryotes. GC content is 30 to 70%. Not sorry, 70%. It is from 30% and it is less than 70%. GC content, G stands for, can anybody say? G stands for C, cytosine, you know. There are the four. What are the GC and A, E,
And thyme.
thymine?
Yes, sir. Thyme.
Okay. So GC content is, it is the number of moles of G and C, which is expressed as percentage of total number of moles of four bases, all G, C, A, T in a DNA sample. Okay. So your four, this is known as base pair of what? It is GC. Base pair is not A.
Yes, sir.
Yes, sir.
So GC mainly it is found. So based on the nowadays GC content also the classification or and also the identification of bacteria is done. Identification. That means a particular species or may not be specific, but at least at the general level, okay, it can be identified and confirmed based on the GC content. That means if this much GC content is there, suppose 55.55, so it will be 66.7, it will be xanthomas. Got? Okay? So classification and identification is also done based on the GC content. Then morphology, come to the morphology. Cell wall. So cell wall, it is rigid, semi-permeable wall. That means semi-permeable means selectively permeable. Okay, it doesn't allow everything to get inside, and it does not allow to pass through it. Okay, all the things. There is semi-permeable wall, 10 to 25 nanometer thick, surrounding bacterial cell. I'll give you the photograph of a typical bacterial cell at the last of this. It is in the last slide. Okay, you'll get. Because in examination, there is every possibility that question may come, and definitely it will come to draw a systematic bacterial cell. Bacterial cell or typical bacterial cell. Okay, showing these different organelles. Then peptidoglycan or mycopeptide, that means both these is together known as murin. So cell wall is composed of peptidoglycan and mucopeptide, and not cellulose. Cellulose is present in
Plant
where?
cell.
Plant cell.
And in fungi,
Chitin.
Chitin.
chitin. Chitin.
And
Pectin.
acetyl glucose.
Yeah. Pectin is present in
algae,
yes, sir. You'll get in the
Algal cell wall.
algal in case of plants, in fruits, pectin. Okay. So this it is your very typical bacterial cell. It is composed of peptidoglycan and peptides, which is not present in like your fungi or like viruses or in plants. So it gives what is its function? It gives strength and protection to the protoplasm. Being semi-permeable, it plays role in control of material exchange between internal and external environment of cells. Okay, and it also have a role in the multiplication of cells. Cell wall, because first cleavage starts at the cell wall only. It also vital role in multiplication of the bacterial cell. Second is capsule. It is extracellular material, consists of polysaccharides of glucose. It is slimy, gelatinous nature, covering the bacterial shell. So these capsules are sometimes they are portable in unfavorable condition to tide over the unfavorable condition, which cover the bacterial cell. Like if unfavorable condition of your water is there, if there is moisture is there, if there is heat is there, so they use the capsules. Okay. Then plasma membrane, it is thin, 5 to 10 nanometer thickness. Lipid, it is a lipid bilayer composed of phospholipids, glycolipids, and a variety of protein lying next interior to the cell wall of the cytoplasm. Okay. Below the cytoplasm, variety of proteins are lying. Then it is also secondary permeable, and it also controls inflow as well as the outflow of metabolites to and from protoplasm. Okay. Metabolites is of two types. It might be known as primary metabolite. Another one is secondary metabolite. You know primary metabolite and secondary metabolite?
so primary metabolite are those which are found in the original natural form, and secondary metabolite is formed from the primary metabolites.
Secondary are found from the
So primary, sir, or some other.
Secondary you tell?
So by the polymerization,
Secondary is found. Secondary?
so by the polymerization of primary or
You told me already.
yes, sir.
One by one. Secondary tell. Why is it not clear? It is produced by secondary metabolites, plants.
No, sir, bacteria, fungi, or plants also, sir.
No, yes. Okay, but here this organism produces both primary metabolites and secondary metabolites. So primary metabolites are utilized by here. You tell primary metabolites are utilized by
Own self.
yes, good. By you tell
Sir,
by the
primary metabolites are those which are directly involved in growth and normal physiological processes of plants.
good. This is it. And it is utilized by the organism itself.
Yes.
Okay. And secondary metabolites are those. These are mainly the toxic materials.
Sir, secondary metabolites are made from bacteria, plants, and fungi.
No. Suppose if you talk of the organism, the bacteria, so these are the secondary metabolites which are mainly the toxic materials. Okay, which are mainly the toxic materials, and these are not utilized by the organism itself. Number two. Number three. Okay, in the active growth, there are four growth phases of the bacteria or organism. You know, lag, log, stationary, and decline. You know.
Yes, sir.
Yes.
Yeah. So these secondary metabolites are formed mainly maximum in case of your stationary phase, and again followed by decline phase. And primary metabolites are produced in
Lag phase.
maximum production in which phase?
Lag phase.
Which phase?
Lag phase.
Log phase, not lag
Yes.
phase. Log phase, growth active phase. Okay. Yeah. So these secondary metabolites are also exploited for it is utilized as antibiotic against your microorganisms like bacteria, and in case of fungi also. Okay, these are because they are toxic in nature. Then it plays also conspicuous role in sexual reproduction. Plasma membrane also plays very conspicuous, very clear role in sexual reproduction. Okay. Then cytoplasm. These are the gelatinous mass of protein. Then your lipids. Okay, the spelling is incorrect. But today only I made the slide because actually I'm not habituated to teach like in PowerPoint with blackboard and chalk extemporary. Okay, but now for this online I have to make the slides for you. So this is lipids, rather carbohydrates, nucleic acid, salts, and inorganic ions. Okay. So these all are dissolved in water. Either granular, they are either in granular form or in the form of a transparent fluid. All these things like protein, lipids, carbohydrates, nucleic acid, salts, and inorganic ions. It contains most of the enzymes of the cell. This cytoplasm, like your, in case of objective type question, may be asked that which organelle contains most of the enzymes of the cell. So your answer will be cytoplasm. Okay. It receives the nutrients from the culture medium and involved in the metabolism and growth of the organism. Okay. Metabolism and growth of the organism. Then plasmids. These are plasmids in bacteriophage. Have you heard name bacteriophage?
Yes, sir.
Bacteriophage. So, is there any role of plasmids? Can you remember?
Sir, vector.
Plasmid. It is also used as a vector. And plasmid you also find in case of bacteriophage. Is not it? Plasmid. So, plasmids, it is extra chromosomal DNA, autonomously replicating in nature, means is auto-replicating. That is when bacteriophages, once the virus infects the bacterial cell, because these virus don't have their like mechanism of auto-replication. Okay, so through these plasmids they go on replicating itself with the association of the bacterial cells, because this is autonomously replicating in nature, present in closed, circular, and double-stranded form. It carries genetic information between the cells of same species, okay, and even between the different species and genera. So it carries the genetic information, okay, from one generation to the another generation. Then mesosomes, these are extensions of, yes,
So the slide is not changing.
slide because you are not able to see cytoplasm, plasmids, no?
No, sir. Plasma membrane, capsule, and cell wall is visible.
Now it's okay? Now it's okay? Okay.
Yes, sir.
Now it's okay.
Yes, sir.
Any problem you can say. That's okay. Cytoplasm it is coming.
Yes, sir. Yes, sir.
Cytoplasm, plasmid, mesosomes, inclusions. Okay.
Yes, sir. Please carry on.
Okay. Mesosomes. These are extensions of cellular membrane of cytoplasm forming much coiled invaginations. So these are the coil-like structures and it is embedded inside the cell. Okay, invagination. And mainly they are most prominent in gram-positive bacteria. So these mesosomes are less. It is less found in case of your phytopathogenic bacteria. Phytopathogenic bacteria. Why? Can you say anyone why it is found very rarely or in less proportion in case of phytopathogenic bacteria? Why? It is most prominent in gram-positive bacteria. Why? Why? Mesosomes are less found in case of
negative.
Forming the scabies, potato scab disease. So, what is your name?
Aniket.
Aniket.
Yes, sir.
Oh, CR. Hey, you are the tall boy in the hostel. I made you CR. Isn't it?
Sir, it was for I was not the CR. Mahindra was the CR.
No, Mahindra, but you are there.
Yes, sir.
Two boys like me. Isn't it?
Sir, but for second sem, all among we selected.
Listen. When you are in the hostel, okay, under meet you, hostel for you.
Yes,
My concern is so that I can remember. Well, why? Anyway, next is it plays important role in respiration of anaerobic bacteria. So in case of anaerobic bacteria, which survives in absence of your oxygen, so it plays a role in their respiration. Okay. It also coordinates nuclear and cytoplasmic division during band diffusion. Okay. So it just acts like a bridge between the nuclear and cytoplasmic division, okay, during the band diffusion. It helps in the synthesis of cell wall. Okay. And inclusions, granules, is composed of both organic and inorganic substances, generally insoluble in nature. Okay. Organics like your carbohydrates, lipids, pollutants, these are organic in nature. Then inorganic like sulfur, calcium carbonate. Okay. So these are generally insoluble in nature. Now slide has changed.
No, sir.
No, sir.
Slide has not changed.
No, sir.
No, sir.
Now it is changed.
Yes, sir.
Yes, sir.
Okay. So what is the first point you tell? Number 8.
Nucleoid.
Nucleoid.
Nucleoid. Okay. So now, nucleoid it is considered as the nuclear area of the bacterial cell wall containing circular DNA molecule and thought to be physical basis of heredity. It is thought to be the physical basis of heredity. Okay. Then it is also considered to be the essential agents in metabolism, growth, multiplication, and the hereditary transmission of characters. And it serves as reserve food material. Okay. It also serves as reserve food material. Then ribosome, as I told this ribosome, one is 70S, one is 80S. Where 80S is present?
Eukaryotes.
Sedimentation coefficient, how it is determined?
By centrifugation technique.
Okay. How? Suppose what the units in centrifugation? What is the unit of centrifugation?
Sedberg unit.
Sedberg unit. Oh, so it is rotated at, I think you might be knowing, RPM. RPM, you know?
Yes,
Revolution per minute.
Revolution per minute.
So this 70S, what is this? That means at a particular RPM for a particular time, okay, in a particular your concentration is also very important. When these bacterial cells are made centrifuged, so that means this is the 70, the unit, okay, that value at 70, it gets the sedimentation, pellets. Above will be supernatant, below will be in the cuvette or in the centrifuge. I think you might have seen centrifuge. Centrifuge equipment, have you seen?
Yes, sir.
There are tubes, isn't it?
Yes, sir.
Yes.
So I think you might have seen your common centrifuge, maybe 1000 RPM like that. So there are this centrifuge, cooling centrifuge, because when you rotate or you give the RPM, okay, like 10,000 RPM, so it gets heated. So bacterial cell will get killed, it will be natured, okay, or like DNA extraction or like that. So it is ultra centrifuge, high speed, and in the cooling character. So when it is rotated there, so 70 is the value that the pellets get set down in the cuvette or in the centrifuge tube. Okay, that is the value. So this ribosomes also it has the role in protein synthesis. Then hemorrhage is pili. Okay. So these pilis are also known as sex pili. Okay. So these are superficial appendages made of protein, smaller and narrower than flagella. So sometimes under specific staining, through the specific staining, when it is done for the flagella, sometimes one may get confused with the pili also of hemorrhage. Okay. So, but these are smaller and narrower than the flagella. It is present in great number. Okay. It helps in adhesion of bacterial cell during conjugation. Conjugation is what type of reproduction process? Sexual or asexual?
Sexual cell.
No. Who told us? It is sexual. Conjugation.
Okay, sexual cell.
Okay. So it also helps in adhesion of bacterial cells during conjugation for reproduction. It also helps in adhesion to the water surface. Okay. For better aeration, because mainly these bacteria, okay, they do not produce the enzymes. So when they attack on the host plant, so it depends on the natural openings or wounds, and it enters. One of the main mechanisms during a rainy season, okay, it enters through the film of water, okay, splash rain. So what happens when it enters? So it get after entering the plant surface or outside the plant where there is water, so it gets adhesion, okay, with the water surface for better aeration. In aeration also it gets, and it moves also, okay, through water. Last, and that is 11th one, that is the flagella. So, size is 120 to 150 Armstrong in diameter, 4 to 5 micron long, okay, and it is cylindrical, hollow, okay, hollow strand made of protein molecule. So this flagella are mainly used for by the bacteria for what purpose?
Locomotory.
There are different type of flagella. Do you know in case of bacteria? Monotrichous, peritrichous, yeah. So based on this, yeah. So based on this flagella also, there is classification is done. Okay, classification of bacteria. Based on the arrangement of in the next class 17, based on the arrangement of the bacterial cells also, it can be classified. Can you say how it can be classified based on the arrangement of the bacterial cell? Can you say Coccus, Diplococcus, Yersinia? You read or not?
Yes, sir.
Okay. So these things I will be teaching in the next class. So next slide, I will show you the ultra structure of this bacteria, which is taken through the electron microscope. Is it visible?
Yes, sir.
Yes, sir.
You see, in examination, you may be asked to draw and write. Okay. So, electron microscope, any idea do you have? Have you seen electron microscope?
No, sir.
No, no one has seen.
Sir. Compound only, sir.
Compound microscope. Where? Dolly?
In my school.
Achha, okay. So you have observed anything through compound microscope?
Yes, sir. Chick cell, onion peel,
Oh,
and blood cell, and and sir, DNA extract.
DNA?
Those
DNA how you
some parts.
DNA how you observe with compound microscope?
Some parts, sir. Like when ethidium bromide
DNA
given.
how you observe?
No, sir. Some parts, sir. Then when white thick portion is filled in chilled ethanol, we can see.
DNA. For DNA, you see, my dear, for DNA electrophoresis, through gel electrophoresis, you have to see against the ultraviolet light, UV light. Okay.
Yes, sir.
These are the bands. They are the bands, DNA bands. Okay, you might have observed something else, like meiosis, mitosis. Okay, also in school, onion growing tips. Okay, you might have
No,
observed.
सर जब सर इथिडियम ब्रोमाइड डालते हैं सर वो चिल्ड इथेनॉल के साथ तो कुछ व्हाइट कलर का सा एक्सट्रैक्ट निकलता है उसको जो स्पूनिंग करते हैं तो वो उसी को हम लोग देखे थे बस कि क्या होता है
मतलब crude, not DNA.
yes sir
Not DNA. You cannot tell it DNA. Okay, it
Yes,
is
sir.
crude. It is
Okay,
crude. क्या चीज़
वो कुछ ग्लास का रड टाइप का था तो उससे निकाले थे
है? क्या चीज़ से? Plant cell.
प्लांट मटर का था सर मटर को पूरा स्पीच करके उसमें डाले थे
Okay. Crude DNA. Crude.
yes
Okay. Like, yeah. Okay. You crush the leaves also under liquid nitrogen. You have to crush it under liquid nitrogen. Okay. Then you can extract the DNA. Anyway. तो कौन सा स्कूल में पढ़ाई की है?
सर जवाहर पुणे
Pune. So, I think you might have seen some of the students like simple microscope, compound microscope, stereo microscope. Stereo microscope, like simple microscope, it will be only 10x. Okay. Like in compound microscope, it will be 10x, 40x, 100x. पता है ये सब थोड़ा बहुत?
Yes.
So,
yes
yeah. So, bacteria are visible under which objective? 10x, 40x, 100x. Where it is visible? Bacteria are visible. Can you say under which objective? Anyone in the school, have you observed the bacterial cell?
No, sir.
No. A chat box, there is no attendance is coming. Why? You have not put any attendance in chat box. Hello, students. Hello.
Yes, sir. अब दिया जा रहा है sir.
Oh, अरे दे दो. So, have you seen anyone during your school in the bacterial cell under microscope? नहीं देखा किसने?
No, sir.
Okay, if you come here, sometime I will make a video. So you see that now you after finishing class, someone is educating. Who is this? Rajiv Ranja. Now he is getting admitted. So, like suppose bacterial cell in stereo microscope is just for observing the your some very minute insects. Okay, some parts of the infected parts of the leaf like that. Then simple microscope only 10x you can observe your just slides bacteria and fungi, but bacteria not very clearly. Then for observing the bacteria, actually compound microscope is needed. Okay. First, after preparing the slides, it is focused under the 10x. Then subsequently, after focusing the 10x, it is focused under the 40x, but not visible very clearly under the 40x. So it is observed under the 100x objective. Okay, 100x objective, and this 100x, when you put the bacterial cells under 100x objective, so you cannot observe as such. In the your objective, you have to put the immersion oil, like cedar wood oil. Cedar wood oil, you have to put a drop over the slide which you have made. Over that one drop of oil is kept. Then only the slide is under the 100x. So under 100x, at least you can observe the shape of the bacteria, whether it is round or it is your rod shape. So these bacteria are also classified as gram-negative and gram-positive based on its cell wall staining. Is not it? Cell wall staining. You know this cell wall staining, why it is known as gram-negative, some of the bacteria, some are bacteria known as gram-positive.
So gram-negative bacteria do not retain the stain when we wash with alcohol. But if we wash with alcohol gram-positive, then the purple color retention.
This purple color is due to which stain? What is the name of this stain? Safranin.
What, sir?
Safranin. Is it?
Yes.
Its name is safranin.
Yes, sir.
Okay. So this is why it is gram-negative, gram-positive, because this is based on the charge present in the cell wall of the bacteria. Okay. Then what is the difference between stain and dye? Mainly we are telling it is stain. So what is the difference between stain and dye? Difference between stain and dye? Find out, okay? To find out what is the difference between stain and dye. Okay. You should know. Anyway, so this is the thing. But from the compound microscope, okay, you cannot visualize, or you cannot see the organelles present inside the bacterial cell. Okay, only the very minute. You can see it in like round shape, like dot-like structure or rod-like structure. Inside you cannot see. Even suppose virus is not visible under the 100x also, because it is more smaller than the bacteria. Okay, and if you see, in terms of size, if we say like bacteria, virus, okay, viroid, then you fungi, then you algae, okay, then protozoa, or leave it like fungi, bacteria, virus. Which one is the smallest one? Because this is your very basic. Which is the smallest one?
Virus.
Virus. Then next,
Bacteria.
Bacteria, then fungi.
Yeah. But all these are unicellular, is not it? Okay. So through electron microscope, it is not present everywhere. I think it is not present in our state also. Okay. So in the very good labs, some labs, universities, it is to the Pantnala. Okay, you can see the animal science, your electron microscope. So for making the slides, a compound microscope like that, it takes your minutes. Okay, 5 minutes, 10 minutes, 20 minutes like that. But for preparation of the slides for this electron microscope, okay, it takes days, 1 day, 2 day, and it doesn't look like a microscope. Okay, it is kept in very big room, 1 or 2 room. It is focused like it is like somewhat like X-ray machine, not like your X-ray machine. It seems like it doesn't look like your microscope, and things can be visualized in a monitor. In that electron microscope, you cannot observe, or it is not observed through your eyes, like shown in case of compound microscope or simple microscope. Okay. Then these are the things. Any doubts from today's class?
No, sir.
You are given all these in chat box.
Sir, but sir, one query, sir. What is what was the difference between conjugation and transduction?
Oh, transaction. Okay. These are the different your reproduction process. Okay. Conjugation is mainly it is the attachment like one positive, one negative. Okay. Attachment of two bacterial cells. Transduction is somewhat also similar. So when I will be teaching you that, I think you see your syllabus. Reproduction is also there in your syllabus.
Yes, sir.
Yes, we'll deal in the detail. Where it is? Which serial number? Yeah, next only. Next only. Basic methods of classification of bacteria and molecules and reproduction. Okay, in brief we discuss. Okay.
Yes, sir.
Conjugation. Yes, tell. So this CR Rohit, na CR?
Yes, sir.
Rohit Datta from next class, next Tuesday. We'll just send the link in the group. I'll join. Okay.
Okay.
If I could have like physical, if I could have done class, so these 2 classes is gone for this, what I have today taught. But it seems that in online class, okay, many things can be completed in a very short time. So I prepared the slide for this only, so that I can show you. Okay. So I thought that it will take time, but actually it happened. Anyway, you require notes also.
Yes, sir.
Notes also required. So I'll be giving you later on. I'll upload in the site notes of this.
Okay, sir.
Anything else? Yes. All have joined attendance. How many have joined?